might imagine, could also happen to repair an AP site that results from a spontaneous depurination or depyrimidination event. And so the way this is processed is first an AP endonuclease makes a cut in the DNA backbone 5' to the AP site. That leaves you now with a 3' hydroxyl, and then on the 5' end you have a deoxyribose phosphate. So basically the sugar and the phosphate with no base, right? This then can be removed by an enzyme called a lyase that specifically removes this deoxyribose phosphate moiety. And now what you're left with is a 3' hydroxyl and a 5' phosphate. Those should sound kind of familiar to you because, of course, DNA polymerases really like to have a 3' hydroxyl. And you have a 1 nucleotide gap. So you have all of the material that you need now to fill in this gap where the lesion used to be, because DNA polymerase can insert the appropriate nucleotide, and then you would be left with a nick, again with a 3' hydroxyl, now adjacent to a 5' phosphate, and that's a substrate for DNA ligase, which can then re-ligate the nicked strand and seal it so that you now have a continuous strand. Okay, so let me fill a couple of gaps in here. So the product of the glycosylase is an AP site. So what the glycosylase does is it removes the damaged base, but just the base, not the sugar. So an AP site results. The AP site is then a substrate for AP endonuclease, and that cleaves on the 5' side of the lesion, of the AP site. This now leaves you with this setup where you have a 3' hydroxyl, but the 5' thing is not an appropriate base. It's just this deoxyribose, ribose phosphate moiety that has no base, so you obviously got to get rid of that. You can't have any basic sites in your DNA. So that's performed by this lyase, which then liberates the 5' DRP. And now you have a 1 nucleotide discontinuity in the DNA chain, so there's a missing nucleotide. So that's a nice substrate for DNA polymerase, and then DNA ligase can seal that up. Okay, so what I'll look at next is I'll show you a little bit of detail of the glycosylase step. Okay, so here's what the glycosylase reaction looks like, and what I'm showing here is the damaged base in this case is uracil in DNA. And then what the glycosylase does is it catalyzes hydrolysis of this glycosylic bond between the sugar and base, liberating the free base and this abasic site within the DNA chain. So the first thing we do is we cleave this. Cleave this bond between the deoxyribose and the damaged base. You can, I hope, appreciate that this is it's the same reaction as we saw in the spontaneous case of either depurination or depyrimidination. It's just that now it's catalyzed by an enzyme. And then the result, of course, is this abasic site and a free base. does uracil get into DNA? Yeah, exactly. Deamination of cytosine, right? So, so deamination of cytosine happens at a pretty high frequency, and so cells expend quite a bit of energy to keep uracil out of the DNA because this deamination is quite frequent. So it's not really clear. I guess you might wonder, well, if cytosine gets deaminated to uracil all the time, how come we don't use uracil in DNA? How can we use T instead of U? And one way of thinking about it is that if you don't normally have uracil in DNA, then you can recognize when the uracil in your DNA is, all the uracil is basically going to be deamination products of cytosine, and so you know to get rid of it. This is all entirely hypothetical, but you can imagine there would be a little bit of a conceptual problem if DNA chains naturally contain uracil and then they get a certain amount of uracil arising from deamination of C. How's the repair system going to tell the difference between bad uracil and good uracil?